Morning, everybody. It's a pleasure to be here today and to be here on behalf of what we call our champions, patients with serious disease that we are addressing at Atara that are daily fighting against their disease. And we call them our champions because we're here to support these champions, to find solutions for them to better fight their devastating diseases. So I'll let you first enjoy your forward-looking statements to make sure you are all clear in your mind about what we're going to discuss in a minute. But let me summarize what we're doing at Atara in saying that we're here to build an innovative, off-the-shelf allogeneic T-cell platform that is relying on Epstein-Barr virus or EBV-specific T-cells that are coming from healthy donors that then we manufacture and prepare as product. And we could, in some cases, apply next-gen CAR-T technologies to these cells to make an allogeneic CAR-T, or we can have EBV-specific T-cells without a CAR just to address EBV-specific disease. We do that through our state-of-the-art manufacturing, not far away from here, and we also match then the appropriate cell for the appropriate patients to allow for safety and efficacy of these cells. So why EBV? Mainly because, as you all know, this is the original oncovirus in all aspects. But it's also a particular virus that is associated with many diseases. Some of them are cancer, especially PTLD, post-transplant lymphoproliferative disorders, other hematologic malignancies, some other type of solid tumors like NPC, or some gastric cancer that are linked with EBV. And we believe as well that there are some autoimmune disease, particularly MS, where there is a particular association with EBV there. Now, these cells, we believe, are quite interesting as an allogenic platform in a sense that not only EBV is present in most of this room, 95% of us here, I guess we're all over the age of 40 or nearly, are really infected by EBV. So we are carrying in a latent way EBV there. But also, when we have these cells, you don't need any gene editing or HLA editing for these cells to be able to be used in patients very safely and very effectively. And the risk of GVHD, in fact, may be reduced by the TCR specificity that we are able to make in these cells, in this treatment there. So these cells, we believe, and we will show progressively some data on that, may be immunologically privileged in a sense that they can expand, they can traffic, and they can persist, and there have been more and more evidence to accumulate on these. What we do with these cells is really to match then in a double way with HLA and the antigen match to allow for recognition only when there are these two elements present there on the EBV antigen presenting cells. And this is applicable already to our first product called Tab-cel or Tab-cel, which is addressing EBV-associated cancers and has already obtained an FDA breakthrough designation and EMA prime designation for PTLD, the first disease that we're addressing with that product. PTLD, for those of you who don't know it, it's a very aggressive, deadly, rare cancer with no approved therapy. And even if rituximab or rituximab plus chemo are being used in practice after that first line of treatment, there is absolutely nothing for these patients. They are relatively young patients, and at the same time, if they relapse or refractory after first-line treatment, they die within a few months. I mean, the median life expectancy is just 1 month in PTLD linked with HCT and 3 to 12 months in PTLD linked with SOT. So very, very serious disease indeed. We've developed Tab-cel, and we have already results showing that Tab-cel there in phase 2 and in compassionate use has been able to show very high level of response rate, between 50 and 80% response rate. And once you are a responder to the treatment, at 2 years, your overall survival is over 80%. So significant level of response, significant level of durable response there. At the same time, safety is quite acceptable in a sense there are few treatment-related serious adverse events, no CRS observed so far in clinical studies, no treatment-related mortality, and it's pretty easy to use. It's like a biologic in many aspects because you don't need any pretreatment. It's a 5 to 10 minutes IV infusion, and then you just need 2 hours monitoring in clinical trials. And we are able, right now, that's what we do every day, to deliver within 3 days to patients the treatment that is appropriate for that particular patient. So beyond that first indication, which is PTLD, relapsed refractory PTLD, which is now in phase 3 development, in HCT and SOT. We're also developing Tab-cel in NPC, nasopharyngeal carcinoma, in combination with PD-1 blocker. And we're also going to start next year a multi-cohort study to address other type of EBV cancer. So very exciting, very advanced program that allows us to deliver to patients what they might need. Now, multiple sclerosis is a very different story, but it's also, we believe, related to a particular association with EBV there. There have been more and more evidence accumulating that multiple sclerosis, particularly in its progressive form, is being associated with EBV in a sense that the loss of EBV-specific CD8 T-cells is correlating with the MS disease progression. And there have been an increased risk of MS when you've had an active infection with EBV when you were younger there. So I don't want to bother you with a lot of science there, but clearly we believe that this is linked with the fact that if you lose, for genetic and environmental reason, this activity of these CD8 T cells addressing EBV B cells, you might then have that issue of multiple sclerosis developing and aggravating due to the autoreactive B cells that are present under control in the brain and in the periphery. So what we're doing here is really to replace what is missing, we believe, in multiple sclerosis, which is very active T cells, CD8 T cells that can act against these EBV cells and control the disease. And our first product, ATA188, allogeneic product there, is already in a clinic, and we presented recently at ECTRIMS some encouraging first data on safety in our dose-ranging study. All the 4 cohorts were well tolerated, no CRS, no GVHD, and these are fully immunocompetent patients. So they are not immunocompromised patients. So it was very reassuring, even for a platform, to show these. And then we start to see some signal of efficacy. We're not saying it's yet the final answer there, but it's going in the right direction, and going in the right direction in terms of clinical improvement or no clinical decline in cohort 2, which was the highest dose. We just have 2 cohorts right now in terms of 6 months data, and cohort 2 shows no decline, no stability. All the patients had some level of clinical improvement with some type of validated scales in MS. So very encouraging, very early, but very encouraging. And our aim is really to progressively build evidence to demonstrate that we may have here, with ATA188, an optimal treatment for progressive MS patients in terms of being well tolerated, ability to reverse disability progression, penetrate the CNS, target specific B cells and plasma cell, and address the underlying biology there. Very exciting time indeed. Now, last but not least, our next-gen CAR-T program is really around developing first autologous proof of concept with autologous CAR-T on innovative targets, and then moving that to our allogeneic platform with the EBV cells in collaboration with leading academic teams there. What we have already in our hands is a great collaboration with MSK, where they presented in June at ASCO some encouraging results of a mesothelin CAR-T. Here, the first generation autologous mesothelin CAR-T, where in patients with advanced mesothelioma, the overall response rate was 63% in these patients, with 3 CR as evidenced by the investigator, and 80% overall survival at 1 year, which compares very well with recent data presented at ESMO with PD-1 blockers. In that particular setting, we use the CAR-T intrapleurally in combination with PD-1 blocker, but the level of results so far are very encouraging that there might be something very unique there. And certainly the safety has been very well accepted, with good tolerability there, no particular issue in terms of toxicities higher than grade 2. And certainly we believe that our unique scFv that binds to mesothelin only in cells that are highly expressing mesothelin, hence cancer cells, is playing a role in this good safety of the product. Now what we're doing now is next year we move to the clinic with a second generation autologous CAR-T that you have here on this graphic, which is leveraging a novel co-stimulatory domain developed by Michel Sadlin at MSK, which is called 1XX, which we believe will have not only improving the ability to improve persistence, but may also be more physiologic in the way it's functioning in addressing the tumor cells there. And then we also are incorporating a PD-1 DNR to avoid having to combine with PD-1 blocker. And that PD-1 DNR is a decoy, basically, that will allow for intrinsic checkpoint inhibition and hopefully unlock the solid tumor microenvironment immunosuppression there. So that is an autologous CAR-T that we are going to with in the clinic next year. And of course, we're already working on the allogeneic version of that CAR-T. And this is in the preclinical stage and will come to the clinic in the next few years. In parallel to that, we are developing a CD19, typical CD19 allogeneic CAR-T at the preclinical stage. And the idea here is to prove the concept of our platform for allogeneic CAR-Ts, where we just take these EBV-specific cells with their TCR and HLA matching that we control and know well from our current product being in a clinic, and then we add a CAR, and then we have a CAR-T which is truly allogeneic in its own sense. So to summarize, we have here, we believe, a potential to transform the life of many, many patients with serious disease based on not only our three strategic priorities, Tab-cel, the multiple sclerosis program, and next-generation CAR-T, but also all that being underlying and supported by our platform of allogeneic T-cell immunotherapies, leading academic collaboration, and world-class T-cell manufacturing that is in-house manufacturing in Atom, not far away from here in Thousand Oaks there. So on behalf of all the patients that we believe are true champion, I would like to thank you for your attention and think that these patients deserve better treatment, and we're here all together in this room to bring this treatment to these patients. Thank you very much for your attention.